Although neuroscientists often shy away from the complexity of it, a big part of what motivates society's interest in studying the brain is that we all want to know what makes us, us. We're very self-interested, and we have a deep feeling that we are all very special and unique. And I don't know if we're all special, but definitely science is showing that we are pretty unique. We discovered the gene, and Richard Dawkins told us that that was everything. Then we mapped the genome, and we discovered that indeed we do all carry individualized instructions on exactly how to build us. But genes can't be everything. After all, identical twins have the exact same genome, but they are different people. They're shaped by the different experiences of their lives. So then in 2010, Sebastian Sung took a further stab at it when he stood in front of a TED audience and got them all to chant, "I am my connectome." So the connectome is a unique set of all the neural connections between every neuron and every other neuron in one person's brain. It's got staggering complexity. There are 100 billion neurons, and each of those neurons has about 10,000 connections. So that means in total, there's going to be 300,000 times more connections in one person's connectome than there are letters in their genome. So far, it hasn't been possible to map the entire connectome. It's horribly difficult, and even if you manage to do it, you just have one person. So you would never be able to map more than one. And not only is it very difficult to get at, it's also a moving target. With every memory that you form, you're going to create new connections and delete old ones. And how stable it is or how dynamic it is an open question. It has to be at least somewhat stable, of course, because if it didn't have some stability, then how could you ever claim to be a self that persists through time if the whole thing is changing, changing, changing? And as we talked about last week, the brain seems to like keeping things stable, and it doesn't even allow for new neurons to be formed in that effort. So although it's been impossible to map a full human connectome at the level of individual neuron-to-neuron connections, researchers have been enthusiastically pursuing the mapping of the connectome using fMRI, or functional magnetic resonance imaging, for about a decade now. And the way this technique works is fMRI is sensitive to blood flow fluctuations in neural tissue. And the assumption is that blood flow fluctuations are correlated with changes in neural activity. So they get these blood flow fluctuation measurements from many different areas of the brain, and they ask, are there places in the brain where we see correlations such that one location and another location have kind of the same pattern of blood flow fluctuation? And the assumption is that if they've got the same fMRI signal changes as each other, they've got a good correlation in their fMRI signal, then those areas are likely to be connected and likely to be functioning together. And then if you build up a correlation network of all of the locations in the brain by all of the locations in the brain, and you calculate the connectivity strength in this way between every location and every other location, then you can get a pattern of an individual's whole brain connectivity structure. Next, researchers ask, now, is it possible to take this whole brain connectivity structure collected from one individual and compare it across a whole range of individuals and identify the one person who produced this particular connectivity profile with good accuracy? Is it possible to use this connectivity profile like a fingerprint? And it turns out, yes, it is. Back in 2015, a paper lead authored by Emily Finn published the first effort at trying to do this sort of neural fingerprinting, and they found that it was possible to use these connectivity profiles to identify individuals with 97.5% accuracy. Absolutely insane. The very first time the technique is applied, 97.5% accuracy. Man, imagine a future sci-fi world where there's no privacy anywhere, because forget hiding from facial recognition technology, which can be overcome with makeup or plastic surgery at the most extreme. How could you ever hide from brain connectivity profiling, brain connectivity recognition? Of course, that sci-fi scenario can only ever come to pass if we have stable connectivity profiles. If we're just whirlpools of changing connectivity with no claim to stability or self, then it's not going to be possible to identify us. We'll have bigger problems because we won't know who we are as our connections change constantly. But it wasn't clear if these connections were stable. So a few years later, in 2020, a team lead authored by Corey Horian published a paper in which they looked at scans that had been collected as much as a year apart, whereas the original study that we just talked about only collected them one day apart. And what Horian and his colleagues found was that you could indeed still identify people when the scans were taken a year apart, just as you can identify someone in pictures that are taken a year apart. But the accuracy wasn't quite as good. They found that they could identify individuals between 80% and 95% of the time, depending on the exact data set they were looking at. So it's still pretty good. But then the question now arises, if we've got these stable connectivity profiles in our brains, and they persist across at least a year, when do they develop? Are they there when we're born? Do they develop as we age? Where do they come from? So to answer these questions, a team lead authored by Dan Hu used fMRI collected from infants who were as young as only 16 days old. They took two different scans that were between 6 months and a year apart, and they asked, just as these other studies we just talked about asked, can we identify individuals from the scans that are taken this 6 months apart? Now, you might think that with the amount of development that goes on in the first couple of years of life, there's going to be no way. The brain's changing so much that there's nothing that's going to be static that you're going to be able to use to identify individuals. But it turned out, somewhat shockingly, that they could. Hi there, I'm Adam Dede, and welcome to a Neuroscience Journal Club, where I bring you a weekly discussion of a recently published paper from the world of neuroscience. If you're liking the content, please don't forget to like, subscribe, leave a comment down below. All right, so let's get right into it. In this graph, you can see that the age at which the participants were scanned, the blue dots indicate when they were first scanned, and the red dots indicate when they were last scanned. As you can see, most participants were scanned for the first time in their first 200 days of life. So the question the researchers were interested in was, would they be able to take the first scan of a baby and use that scan to identify that particular baby out of the full set of second scans? Basically, can they use a baby's connectivity profile to identify that particular baby? Now, in previous research, it's been shown that different regions or networks of the brain have different individual levels of uniqueness. So they parcellated the brain out into different functional networks. You can see in red and yellow are the dorsal attention and frontoparietal networks. These are important for higher cognitive functions like action planning, goal setting, and working memory. Then in green, you can see the default mode network. These areas are important for daydreaming, remembering the past, imagining the future, and thinking about the feelings of others. In navy, you can see the visual cortices. These are important for vision. In medium blue, you can see the limbic areas of the brain along the medial temporal lobe and wrapping around the rostral corpus callosum. These areas are important for emotional processing. And then in light blue, you can see the somatosensory cortex. These areas are important for bodily sensations and muscular actions. Finally, in purple, you can see the ventral attention network. These areas are also important for directing attention, but towards more object or conceptually oriented targets compared to the dorsal attention network, which orients attention more spatially. Now, even with this parcellation, it turned out that identification was quite poor. As compared to the 80 to 100% identification rates that we talked about in some of those prior studies that were done on older individuals. As you can see here, network number 1, which was the dorsal attention network, seems to have yielded the best identification rate, but it was still only around 40 to 50%. For comparison, here's an analogous figure from that paper I mentioned by Corey Horian. Again, different brain networks are indicated along the x-axis, and identification rate is along the y-axis. The different colors indicate different data sets that were examined in this study. The key difference in this figure versus the one we just looked at is that the participants in all four of the data sets being examined here were not babies. The data set in blue was composed of adolescents between 10 and 20 years old. The data set in red was composed of young adults in their 20s. The data set in green covered a wider age range from teens to 40s, and the data set in purple was comprised of older adults between 50 and 80. So taking all of this together, we can see that there's a general pattern that younger brains are less identifiable by their connectivity profiles than older brains. Now here, I've taken these identification rates from the two different papers and just grabbed the ID rate for the best network in each data set. As you can see, it appears that for babies, the identification rate is much lower than for older children or adults. And there seems to be a trend such that people's brains get more and more uniquely stable as they age. Now, Hugh and colleagues didn't stop there. They realized that there was no strong a priori reason to assume that the parcellation of the brain that they had used would necessarily yield the best possible identification. So instead, they chose to follow the brain's inherent variability, which, as we've seen before, is really a great way to try and get a handle on what it is that's going on in there. What they did specifically was they looked at which connections had the largest amount of variability between different infants. And they assumed that, okay, if this particular connection has a lot of variability across different infants, it's likely to be one that allows for the best possible identification of particular infants. And this is represented here in these two correlation matrices. So the strength of the self-correlation increased when correlations were calculated only using high variance edges. To see this, let me orient you to the figure. Each pixel of these heat maps is showing you the strength of similarity between the connectome map of the indicated session 1 brain scan on the left and the session 2 brain scan on the bottom. The diagonal line that cuts from the top left to the bottom right represents the strength of similarity between the connectome maps created from two different scans for the same subject. Identification of individuals depends on the diagonal being stronger than all of the off-diagonal similarities. In the plot on the left, you can see that the diagonal is visible, so identification is possible, but there is a lot of strong similarity off the diagonal as well. By contrast, on the right, you can see that the diagonal pops out much more strongly. In other words, by selecting only a subset of the connections that were most variable between individuals, Hugh and colleagues were able to increase the contrast between individuals and make identification better. So this graph shows you exactly how many connections to use to get the best identification. On the x-axis is the threshold of what proportion of connections to disregard. So a threshold of 0 means that all connections are used. A threshold of 1 means that no connections are used at all. As you can see, the best detection occurs when 95% of connections are thrown out and only the top 5% of most variable connections are used. Okay, so to think about what this graph is really telling us, you have to think about what are the factors that go into good identification. And there's two that you need to consider: uniqueness and stability. And you need both of these in order to have good identification. So let's think about a situation where you have good stability, but poor uniqueness. So let's say that I tell you to go into a crowded room and find my friend Bob. He's the guy with 4 limbs. Well, it's highly stable. Bob's had 4 limbs his entire life, but it's not very unique, and you're not going to have much chance of identifying Bob. Then let's think about a situation where there's high uniqueness but low stability. So yesterday when I saw Bob, he was wearing a red shirt, and that's pretty unique. Not everyone's wearing a red shirt. So I tell you to go into the room and I say, Bob was wearing a red shirt yesterday. Go find him. Well, you got no idea. He might not be wearing a red shirt today. Probably not. So that's a situation where, although wearing a red shirt is pretty unique, it's not stable. So then finally, let's think about a situation where there's high stability and high uniqueness. I say, hey, go in and find my friend Bob. He's the guy with the shamrock tattoo on his neck. Now, it's likely that you're going to be able to find him because he's probably the only guy in there with a shamrock tattoo on his neck, and it's probably not going to change. It's got high uniqueness and high stability. So, thinking about stability and uniqueness then, the question is, do the connections that are being discarded in the identification of infants, do they lack in stability or do they lack in uniqueness? Which of those is making them bad for identification? So here it's important to keep in mind that adding those extra connections in the infants is actually making identification dramatically worse. Look at the graph again. You can see that when you use only the top 5% of connections, you're getting about 80% identification. But when you use all of them, it's way down at 60. So why is it that adding more connections actually makes things worse? And this is very different from what we see in older populations. So here we're looking at a graph from that earlier Horian paper. And you'll remember that for these subjects, using the best possible network to identify them raised identification to between 80 and 100%. And in this graph, what you can see is that when you use the whole brain at once, identification is still up at between 75 and 95%. In other words, selecting the best network only bumped identification up by about 5% in these older populations, whereas for infants, selecting the best possible set of connections is bumping up identification rates by about 20 or even 30%. For an even more analogous example, let's take a look at this graph from Lisa Burge and Daniel Kennedy. who used a technique very similar to that applied in the baby data. Like in the baby data, they asked what would happen if they took only the most individually distinct edges into account when doing identification. This graph's x-axis is opposite to the one we looked at for the babies. Here, 0 indicates using no connections, 10 indicates using only the top 10% of most distinctive connections, and 100 indicates using all connections. The y-axis indicates identification rate. What you can see is that the identification rate quickly rises as more connections are used, and then it goes flat. So this is totally different from what we saw in babies. In babies, we saw that adding more connections actually made things worse. So how do we interpret this? What it seems to be then is that in the adults, adding more connections doesn't hurt us because those connections are stable but non-unique. It's like if I tell you to go find my friend Bob and he has a shamrock tattoo and 4 limbs. Okay, well, telling you he had 4 limbs doesn't hurt your identification ability because it's highly stable. When you go in and search for him, he'll still have 4 limbs, but it's not going to really help you either because so do most of the people in the bar. By contrast, the babies are likely then to have non-stable but highly unique connections. And these are the connections that are causing poor identification. It's like if I tell you, look for my friend Bob. He's got a shamrock tattoo and a red shirt. But he had a red shirt yesterday. He might not have a red shirt today. So yes, the shamrock tattoo is unique and stable, but the red shirt is unique and non-stable. And by adding it in, I'm going to have you searching the room for red when he might not be wearing red today at all. In a similar way, what this tells us then is that the babies have a large number of unique and non-stable connections that are changing over time. And they're changing in fundamentally different ways from each other. So then on the flip side, what about those 5% of connections in the babies that were stable and were unique? Where were they, and how did those contribute to the identification of the babies? In this chord plot, you can see the set of connections that were most diagnostic of individual identification. As you can see, connections between dorsal attention areas were important, and so were connections involving visual areas. For comparison, here's a similar plot of the most diagnostic connections for identifying individual adults from Berg and Kennedy's paper. As you can see, frontal areas shown in the top in reds and oranges are still diagnostic, but visual areas shown in purple are much less diagnostic. This is true if we refer back to adult longitudinal data from Horian also. Here, networks 5 and 6 are the visual ones, and you can see that visual networks tend not to be highly diagnostic. We know, though, that adding visual networks into whole brain identification analyses for adults does not reduce identification accuracy. Logically, then, what this tells us is that the visual cortex connections in adults are stable but non-unique. But in babies, they are stable and unique. This tells us then that babies have fundamentally different visual connections from one baby to another. And over time, through experience with the same world, we all coalesce into having the same set of visual connections across development, which is kind of crazy. What this tells us is that babies are out there like tripping in totally unique ways all the time. And it's only as they get older that they manage to see the world in some kind of a standardized way that's similar to everybody else. If you think about that kind of age-old question, like, is the blue of the sky the same for me as it is for you? Yes, probably, because we have the same set of connections in our visual cortices, but for babies, no, probably not. They are experiencing a fundamentally different set of visual inputs from one baby to another, which is just wild. These individual differences seem to get ironed out pretty quickly, though, because when Hugh and colleagues split their data up by age group, what they found was that for babies between 0 and 6 months old, the visual system was about twice as diagnostic of individual identity as any other system of the brain. But when they then looked at babies who were between 6 and 9 months old, the pattern had already reversed, and the dorsal attention system was twice as diagnostic as the visual system. Now, the visual system was still the second most diagnostic, which is still very different from adults, but you can see already that there's this pattern of developing where the visual system is heading towards the stability that we're going to end up seeing in adulthood. Anyway, that's it for this time. Thanks for watching. Hope you liked the video. Please click the like button, subscribe, comment, all of that good stuff. And if you've been a regular watcher, Please subscribe on Patreon and support the content. Thanks. See you next week.